Pharmaceutical Research Institute in Albany, New York, and he is honoring us by his presentation today, the impact of nanobiotechnology on the future of pharmaceutical industry.
نحييكم بتحية الإسلام. السلام عليكم ورحمة الله وبركاته. It is a pleasure and honor to be here among colleagues and friends, and I want to thank again the organizer, Dr. Amal Bishlawi, Dr. Matar, and Dr. Qari, and the rest of the team who put together this magnificent meeting in our beloved Cairo. I'm going to share with you very briefly key concepts because I can't go over a lot of comprehensive details. I want to leave key messages that stay with you. And the concept here is about nanobiotechnology. We speak about nanotechnology, and without linking it to biotechnology, nanotechnology is an enabling technology. Without biotechnology, I hate to say it, but it's useless technology. So you have to, especially in the medical field, nanotechnology has a wide range of applications in medicine and everything around us. I'm going to focus my attention to nanomedicine. And nanotechnology, without the knowledge of biotechnology and a comprehensive multidisciplinary approach, a team approach that include every discipline under the sun, from a physicist to a chemist to molecular biologist, immunologist, pharmacologist, a clinician are very crucial because they need to guide the translational potential of what we do if we want to make a difference. Clearly, there is a major impact of nanomedicine on the future of medicine. We're going to see a lot of new medicine that will come forward very quickly to improve existing medicine and bring a new innovative medicine to the front to solve key medical problems with unmet medical need. And also it will enable us to approach the road to personalized or to be precise precision medicine, where the idea about one medicine fits all is not really the right idea. We have to personalize and actually give the right dose, the right medication to the patient. And that will be achievable with the knowledge of gene sequencing and pharmacogenomics. Clearly, nanotechnology, I can witness that in the last 12 years in academia in Albany, New York, which is the capital of the world of nanotechnology, and you can check it out on your Google. Just put Albany, New York. It is the capital of nanotechnology. I can tell you the proof of the pudding is the state of New York invested $1 billion to build up a huge hub of nanotechnology, and it attracted every company under the sun in terms of nanochips, the wafer, all of the revolution that you are witnessing in your cell phone and the apps, and the use of the cell phone to manage a patient, and biosensor technology. All of this is being accelerated at that site, which is called Polytechnic Nanotechnology Institute, within a spin-off from the State University of New York. It started with $1 billion investment from New York State. Now it's $15 billion enterprise. There is about 3,000 people working at that site. There is about 250 spin-off companies out of that. So you can look at the economic and the societal impact. And that's what that nanotechnology is all about. You look at the graph here. I'm going to use the cursor so I can, you look at the societal impact. Industrial revolution took 200 years to realize its full potential. Nanotechnology, in terms of the revolution in molecular nanomanufacturing and impact on translational, can take actually 1 decade or 2 decades to get the full potential. So we hope that the Arab world will wake up and realize the potential of nanotechnology by implementing it, not by talking about it. bringing to the front in terms of applications. So there is a huge economic and societal impact that nanotechnology can do in every field in a way that you can, energy, everything. I'm just going to focus my attention to nanomedicine. Why nano? Nano is a small size. In a way, you have many of the molecules, the drug that we use. If you can nano size them, you can increase the surface area. You can improve the physical properties. You can improve bioavailability. But furthermore, you can use a shell to host nano, to host the active pharmaceutical inside. You can put a targeting moiety on the surface of the shell, and then you can put an imaging probe to actually monitor the trip that the nano loaded with the drug is going. And then you can put many drugs, multiple chemotherapy, and you can target them to the surface, to the site of action. So you can improve efficacy and reduce toxicity. I'm going to show you examples of that. Very brief. Traditional drugs usually goes everywhere, but when you nano target the delivery, you can deliver differentially drugs to the liver, to the kidney, to the heart. You can deliver them to the tumor and so on, to the brain. So you can achieve a target localization, and you can achieve a better efficacy and safety. And as I mentioned before, nanotechnology standing alone is not really the way to go. You have to bring biotechnology, bioinformatics. We have to benefit from the pharmacotherapy, the traditional pharmacotherapy, optimize our knowledge about the PK, the pharmacokinetics, the pharmacodynamics, and in the center here, pharmacogenomics. This is how we can, in a way, tackle and use the technology, enabling technology to our advantage. So it's about technology integration. Stem cell therapy can benefit a great deal from nanotechnology because you need nanoscaffold. And you can optimize the functionality of the stem cell. And you can host the stem cell in an environment where they function to differentiate into whatever you want them to differentiate to. Endothelial cell, you can differentiate into osteoblast, and whatever is required. A nanoparticle application in drug delivery, that is one of the most advanced moving forward application. Gene therapy, because many of the protein or the RNA or the DNA that we want to deliver, they are unstable. They need to be hosted in a shell to improve their stability. And you can imaging, MRI imaging, PET imaging, you can improve the quality and the resolution and the specificity, sensitivity of the current technology that exists by using a polyvalent probes on the surface of the nano. That will give you about 10-fold improvement in the resolution. And this is getting close to become reality. There are a lot of products now in advanced clinical trials, and there are products in the marketplace. So this is not really a science fiction. This is a reality. Now, drug discovery, drug development takes a long time to be achieved, high risk for pharmaceutical company. Anything wrong can go in any junction. Moving from that cascade of event, I lived in that life for almost 2 decades. So I learned the in and the out to drug discovery, drug development based on that experience. It is a long process. And the minimum that you can actually get a drug from the bench to the bedside, about 7 years. It can take 10 years, 12 years. And by then you have about 10 years left on the patent life. So the company tried to rush to the market and make the most out of those leftover time. So here in nanotechnology, I can tell you, I do that, that's something I try to, do very well, is I bring a new life to all the products. And I'm going to show you an example of that. And I started the car with 0 mileage. So I get about 20 years life on the product. The only thing I need to do by equivalence. Did I improve the quality, the efficacy and safety of the nano-formulated product relative to the existing product? If I can get that, clearly the FDA will give you the green light to approve it. And you don't need to go to phase 1, phase 2, phase 3, and so on. So indeed, could we this enabling technology shorten the life to drug discovery and drug development? Absolutely yes. You can reformulate and improve the PK and PD. Previous session, I showed that you can make heparin in an oral form. And this goes with low molecular weight heparin. You can do the same thing for insulin. You can do a combination. If you look at the hepatitis C virus, there is a polymerase inhibitor, protease inhibitors, and given in a separate forms. You can actually, create a nanoshell, put the polymerase protease together in under one shell, and you can target the virus. We have a lot of knowledge about molecular signature that exists on the virus that doesn't exist on the human host cells. So I can actually deliver the polymerase protease inhibitor into the virus and shower the virus, and I can cut the dose by tenfold. You can look at the economic impact, the safety, and the efficacy, and instead of say, the best that we can do now is Dacla and Sofosbuvir. If I target it, it takes about 3 months. This can be cut down to 1 month max, and you avoid the relapse. You don't give a chance to the virus to be circulating without it being attacked all the time. You can sustain that delivery. You can do chemotherapy. This is actually highly achievable. You can deliver chemotherapy differentially into the tumor by about 10 to 100 fold. There is no chance for the tumor to think about resistance. Because resistance, when you try to tweak the tumor by exposing them to low levels, they try to adjust the genetic makeup, the multiple drug resistant, the transporter, the G protein, all of that, and upregulate those to just kick out the chemotherapy. But if I just keep showering them with a high level from the beginning till I get rid of them, then there is no chance for resistance. So this can be highly achieved, and then you avoid the side effect of all of the chemotherapy. You can actually do this with antibodies. You can achieve a genomic versus non-genomic and imaging. I'm going to give you a quick example that actually based on my experience, that I'm actually dealing with this. There is a newcomers into the marketplace, antibody against PCSK9. PCSK9 is an enzyme that is in the hepatocyte. This enzyme, some of us are unlucky to have a gain of function and high expression of that enzyme in our hepatocyte, we will accumulate cholesterol because that enzyme degrades the LDLR receptor in the hepatocyte that cycle the cholesterol from the blood and get rid of it. People who are lucky, who have low expression level of PCSK9, if those are homozygous familial hypercholesterolemic, they can take an antibody of that enzyme and they can lower their cholesterol way beyond the statin. Statin has a lot of limitations. Statin actually, over long-term use, multiple years, overexpress that enzyme. The enzyme is overexpressed. So this enzyme is localized in the liver. This is the only thing that exists now, two antibody approved by the FDA, and we took upon ourselves 8 years back to develop a small molecule, solid dosage form, a small molecule, oral compound, that we can combine it with a low dose of statin or by itself. And we can do nanohepatic targeting. We can target the liver where the enzyme is. That's where we are. So I'm going to show you the concept very quickly here. The LDLR receptor on the hepatocyte, it is sensitized. It goes to and it circulates into the cell surface, and then it captures the LDL particle, shunt it, take it, and digest it, and get rid of it. And then the receptor recycle back into the cell surface to take care of other LDL cholesterol. The problem is usually if you have an overexpression of this enzyme, which is PCSK9, also synthesized in another vesicles, and it comes to the surface and bind to the LDLR and degrade the LDLR. So the LDLR is not capable of taking the cholesterol from the blood into the hepatocyte to be degraded. Or it can actually bind and stay with the LDLR cholesterol and get inside and degrade the receptor, so the receptor there is no receptor to be recycled back. So now you have a panel of monoclonal antibody by 2 companies, a third company is coming to the scene, approved by the FDA already, and they do what they are supposed to do. They reduce cardiovascular events They lower LDL cholesterol by 70%. They are good, you can take lower doses. You can bring your cholesterol way back to very low. The American heart is pushing for the lower cholesterol because if we think about, this is atherosclerosis. High cholesterol, you have a risk of atherosclerosis. And then if you have atherosclerosis, that can bring thrombosis when the plaque is ruptured. So I think this is a very good thing to maintain your low LDL cholesterol. So this antibody is moving to the front. The application here of nanotechnology, which I don't have time to go over, this is a separate talk by itself. We made a small molecule, we do the hepatic targeting, and we can deliver those molecules to block either the active site of the enzyme or the binding of the enzyme to the LDLR. So we have two different types of inhibitors. And now we can deliver them into the site. And we can achieve the same thing like a very expensive antibody. Small molecule, like more or less the same molecular weight they like statins. So these are the antibody approved by FDA, and many more are coming. Very expensive, one injection every 3 weeks. The cost will be in the range of $3,000. Maybe for familial hypercholesterolemia is okay. But for regular people with a marginally high cholesterol, maybe it's too much. I don't know. But the market will dictate. Now, the other question, could we convert? And I think I showed that at the other session. Could we enable to convert an injectable heparin GAG, glycosaminoglycan, which is a polyanion, into an oral dosage form? The answer is absolutely yes. So I'm going to skip many of this stuff because we've proven, indeed, if you take low molecular weight heparin here and you make it a nano formulation, this is oral nano formulation of low molecular weight heparin. It doesn't matter which one. This is applied to all low molecular weight heparin. And if you give the free low molecular weight heparin orally, it has 0 oral bioavailability. It doesn't get absorbed. But once the nano shell get into the blood, there is enzyme in the blood that breaks it down, and you get only low molecular weight heparin. Now what you need to do, bioequivalence studies. You do a nano low molecular weight heparin versus free low molecular weight heparin, but you give the low molecular weight heparin subcutaneously, and you give your nano formulated orally. If you can come up with the same bioequivalence, then you can claim all of the indications for that drug without going into this multiple phases. Cancer. Cancer is a big dilemma. You look at, there is a lot of advances made in cardiovascular disease. The risk is reduced by 50%, MI and all of the adverse events. Cerebrovascular with significant advances made, pneumonia and infections are big advances, but cancer from 1950 till now, there is no statistically significant difference in terms of improving morbidity or mortality or quality of life of the cancer patients. So now we ask ourselves, can really we borrow this enabling technology to help us improve existing medication and future medications? Now we have to dig into the differentiation. So we look into blood vessels, normal blood vessels, and the tumor blood vessels. And if you look at the tumor blood vessels, it's very leaky. The vascularization around the tumor is highly leaky vessels, but there is also a certain receptor that's highly expressed, that's not expressed on normal vessels. This receptor is from the family of integrins, alpha V beta 3 integrins, which is highly expressed on the tumor cell itself and on the rapidly dividing blood vessel around the tumor. So now we have good chemists, we can develop a high affinity ligand for the alpha V beta 3 integrin, conjugate it to the polymer, assemble the nano, and we can fill the nano with the chemotherapy, and we can deliver it to that site without it getting into normal sites. Additionally, that target by itself has a major anti-cancer effect. It's an anti-angiogenic, anti-proliferative. So we discovered actually, without even the chemotherapy, we can we can turn on tumor suppressor gene and turn off tumor survival genes. And one of these drugs is going to clinical development, in September 2016 at MD Anderson for glioblastoma and at Cleveland Clinic in pancreatic cancer. And both received an FDA designation for orphan diseases. So the FDA approved the trial to go forward with this. This is what one of the molecules that's going to go forward into clinical development in GBM and pancreatic cancer. And this is the characteristics, in terms of the size and the micrographic, how the particle looks like, very uniform, very homogeneous. This is a classic characterization that you have to provide as part of the monograph. We tested a lot of different cancer types, and this receptor is overexpressed in all of them, and they survive on new vascularization where the receptor is expressed, and we can tackle all of them. But we are starting with the most vicious type of tumors, GBM and pancreatic cancer. And if we can win the battle against those two cancer types, those are easy ones to achieve. So we are actually challenging the status quo by going after the highest risk type of tumors. And the trial is very short, 3 months. If we can extend the life and eradicate the tumor based on MRI imaging and based on markers, then we should be able to go after the other tumors. And these are images to show the data. We published extensively on this. There is about 60 manuscripts published on the concept. We can also, how about if we can fill the shell inside along with the alpha V beta 3 ligand outside, can we deliver cisplatinum into the tumor? Can we deliver other chemotherapy into the tumor? We tested many of these chemotherapies, cisplatinum, doxorubicin, vincristine, Taxol, many of them. And we can show here, if you give cisplatinum, this is the level you get into the tumor. You get about 20 nanogram per gram tumors. If you put it inside the shell with targeting, you get about 100 nanograms, so about 5-fold increase in the delivery. And we have seen this study done in preclinical models, nude mice. We see that we can eliminate peripheral neuropathy. And so this is very important. The side effect is eliminated and the efficacy is improved. And you look at Taxol or paclitaxel in pancreatic cancer, you can see almost about 10-fold in an increase in the delivery. The same concept can be extended using a chelator called DATA, and you can put a gadolinium or copper-64 for MRI or for PET imaging. You can change the probe, a technetium for SPECT imaging, or micro CT imaging. And now the targeting moiety here in that case is, I'm sorry. I'm going to go back here very quickly. I'm getting toward the end. I got it here. All right. I'm going to go here. Okay. So, the other, so we can actually deliver a lot more into the tumor and also imaging-wise, and you can do the two simultaneously together. So you can actually monitor the therapy and by early detection of the tumor. Also, you can follow up the tumor progression by image analysis within the same core of molecules. I just want to highlight here, this idea about one drug fits all, one dose fits all. Look at the statistics in terms of the percentage non-responder for many diseases with many classes of drugs that are conventionally used. There is almost 1/3 of the patients get the most benefit out of the drug, 1/3 get marginal benefit, and the other third get nothing but the side effect. So the future is to use this enabling technology to customize and adjust the dose to the patient based on pharmacogenomic information, which is going to become a reality and easily achievable. So this is what we predict in the next 5 years, the next 4 years. Actually, this was my prediction 5 years ago. It is coming closer and faster, that we should expect, based on the cost of DNA sequencing, that this will be the case for routine checkup, for prevention at the primary care physicians. This is what in the US. So we hope our primary care physician here in Egypt are up to the bar in terms of catching up with the US primary care physician. We can match the tumor to the drug. My goodness, there it is. We can match the tumor. We can also, nowadays, there is an immune oncology, immune therapy stimulating. This is moving like a revolution. And also gene therapy using nanotechnology. Without nanotechnology, gene therapy will never become a reality. So rare disease, like what I mentioned, sickle cell, it's a single amino acid flip that should be easily fixable. That might take some time, 5 years. Biosensor, automedicine delivery, it's coming. Improve healthcare delivery, robotic surgery, biocompatible body parts. It is happening. Tissue engineering is making huge advances. So these are actually is going to improve the quality of the healthcare and the quality of life for us living on this earth. And I just want to finish here by showing my final slide. I showed that in last year, but I like to keep it. We have to be not to be traditional. We have to think outside the box, and we have to envision what can we do to bring a better life to the patients through our research, even clinical research, whatever research we do. You look at the artist, he's looking at the egg, but what is he drawing? The bird. But more important than imagination, or as important as imagination, execution. There are a lot of people who have great ideas, but they never act upon them. With that, I like to thank a lot of people, army of people who actually enable me to come here and speak to you because they provide me with all of this data. And I collaborate with a lot of people in the US and outside the US, and that is the secret of success. If I have to say what is really the secret of success is collaboration, collaboration, collaboration. And that will amplify your effort and bring winning people around you. And so this is a very important concept in life that all of us have to adopt. Thanks so much for your attention.
I would like at the end of this very interesting presentation to thank Professor Musa, and we can throw some questions and then move to the next presentation. Before I do that, I would like to remind everybody that 3 o'clock is the Pan-Arab Hematology Association Assembly meeting, which is open for everybody. It's in the Abdeen room, the one floor up, -3. We are in -4. The meeting will be at -3, Abdeen room number 1. 3 o'clock, Pan-Arab Hematology Assembly, and it's open for everybody.
You mentioned that we can use the nanotechnology for imitating the microenvironment of hematopoiesis, the scaffold for stem cell. Can you explain this for us, please, in short?
This is a separate lecture, so maybe I'll catch up with you afterward.
Thank you so much.
Because the stem cell is a separate domain.
Yes.
It is very huge, and the
Yes,
nanotechnology will have a major impact.
Yes. Second question, yes, you mentioned that immunotherapy together with nanotechnology actually represent huge advances in cancer treatment. Also, we need some illustration of this. I know it's a big subject, but maybe in another lecture or...
No, I just briefly here, I'll mention to you, in a way, these are becoming reality. There are a lot of nano, in a way, chemo, but without active targeting, that are approved by the FDA. There is a conjugate, there is a nano paclitaxel albumin conjugate, is called Abraxane. That's approved in the marketplace. That replaced the paclitaxel in the marketplace. This drug, just think about it, this drug as a nano platform, it has, because nano has huge permeability even by passive transport to permeate the tumor, which is very leaky, and it accumulates by about 2-fold. And that is based, let me tell you the market valuation so people can understand how the market is accepting this. The market for this drug, which replaced paclitaxel, is about $1 billion in the US alone. That's what, and this company, Celgene, and many companies are, and there are many other chemo, but all of them are passive transport, just putting them in the shell. The future, which we are doing here, is to do active targeting moiety. And instead of achieving 1-2-fold delivery into the tumor, I can achieve 30 to 100-fold after optimization. And that will enable us to get rid of the side effect of the chemo. At the same time, avoid the relapse and the resistance that's developed.
Thank you so much.
Thank you very much, Dr. Shakir Musa, for your very comprehensive presentation. And now our second speaker is Professor Dr. Triplo Bush. He is the head of an Institute of Emerging Diseases and Innovative Therapy and Professor of Medicine in Paris University. He is also a visiting professor in Harvard University. He has a lot of publications in gene therapy, especially in thalassemia, and his talk today will be gene therapy in thalassemia.
Okay, thank you very much for the invitation, for the kind introduction. I will try today to review for you the status of our work on the gene therapy for thalassemia and sickle cell disease, two sister diseases. And as you know, the only current curative treatment for thalassemia and sickle cell disease remain allogeneic hematopoietic stem cell transplantation. But a lot of patients, most of them, do not have an HLA-matched sibling donor, and those who do still face the risk of engraftment failure and graft versus host disease. A lot of progress has been made in allogeneic transplantation, especially with the use of cord blood cells or mismatched donors, but it remains, as shown here in a quite recent study in 2011, that the overall survival rate is about 94% for sickle cell, but only 62% for thalassemia, and the disease-free survival is only about 50% for sickle cell disease and 21% for thalassemia. So there's still a lot of work to do in this area of allogeneic stem cell transplantation. Now, gene therapy offers an alternative even for patients who do not have an HLA-matched sibling donor, and in all patients that are candidates, we do not use a conditioning regimen with chemotherapies that are as strong, I would say, as for allogeneic transplantation, because you do not need to induce immunosuppression, and therefore the risk appears lower. So because of all these reasons combined, and especially the possibility of reaching out to patients who do not have an HLA-matched sibling donor, gene therapy is of a major interest to us in the field. Well, there are many approaches to gene therapy. Our colleague spoke about nanotechnologies, but with regard to thalassemia and sickle cell disease, and especially thalassemia, it's important to remember that thalassemia can be caused by many, many different kinds of mutations, hundreds of various mutations along the beta-globin gene locus, and therefore it would be very difficult to receive approval from the regulatory authorities for instance, products that would aim at correcting each mutation, because you need to have as many products as mutations that exist. And therefore, gene addition, by bringing a normal beta-globin gene expressed appropriately in the red blood cell lineage, remains the most logical approach. And for sickle cell disease, it is possible also with the same product, and I would explain to you how, to inhibit sickle cell formation, and therefore with one single approved medication or product to target both all cases of thalassemia and sickle cell disease. So as I explained a minute ago, for gene addition, what is very important is to know where you want to add the therapeutic gene. And logically, if you want the effect to be maintained for the lifespan of the individual, you want the gene to be introduced in the genome of hematopoietic stem cells. Otherwise, upon cell division, differentiation, the material will be lost and diluted out. Now, if you introduce the gene in the hematopoietic stem cells, you nevertheless want the gene to be expressed appropriately and regulated in the red blood cell lineage. You don't want to make globin in white blood cells, okay? And you also want to express it at very high levels, because globin is the protein in the body expressed at the highest level, even higher than albumin. So it's a challenge. Now, in order to do this, we need to choose the right vector. And so far, only viral vectors have ever shown that you can correct a hemolytic stem cell in an animal model, whether it's a mouse, a primate, including humans, of course. Only them have shown the capability of doing so long term. And among the various viral vectors, the ones that have a special interest are lentiviral vectors derived from HIV or other types of lentiviruses. And that's for two reasons. The first is that they are capable as a pre-integration complex to enter through the nuclear pores of the nuclei and therefore integrate the DNA material in the chromosome of cells even when they are quiescent, when they do not divide. And that's very important because when you harvest stem cells, most of them are quiescent, and you cannot really in vitro trigger cell division without losing their essential properties. So that's very important to be able to do so even in quiescent stem cells. And the second property that they have is that they are capable of carrying very complex and quite long DNA structures without rearrangements. And that's because of the splicing machinery they have with the REV, that's the REV protein of HIV, and the REV responsive element, RRE. For those two reasons combined, lentiviral vectors have shown great efficacy and appropriateness in using them for gene transfer to hemolytic stem cells, especially for globin gene disorders. Now, with regard to the type of globin gene we want to use, well, there are many possibilities. The first one would be to use the normal human beta globin gene. However, that would not be effective for sickle cell disease because the normal human beta globin gene is poorly inhibitor of sickling. It acts only by dilution, whereas certain amino acid residues that you find, for instance, in the fetal gamma globin protein are very strong inhibitors of sickling. And what we've done is to incorporate a key amino acid residue at position 87 from the gamma and also the delta globin genes into the adult beta globin gene structure, so that now you have a very strong anti-sickling therapeutic product. And the advantage of doing so exists also for thalassemia gene therapy, because since that globin protein is tagged as an amino acid residue, by HPLC analysis, you can precisely identify and quantify what is expressed from the vector. Otherwise, you would not know what happens if the patients remain transfused somewhat, or if it is a case of beta plus thalassemia. So you see here example at the bottom of HPLC profiles, and you see a new peak with beta 87, what we call beta 87Q, which is the therapeutic globin gene with the tagged amino acid residue, which is also anti-sickling. Now, in terms of what kind of structures we need to accompany the coding region of the beta globin gene, well, we need to use also the introns and the flanking domains, including the beta globin promoter. Otherwise, transgenic animal experiments have shown that you do not get a sufficiently high level of expression, and it's not properly regulated in the red blood cell lineage. But in addition to that, another challenge that occurred was that we needed to add to this key elements of the so-called locus control region made of DNase I hypersensitive sites, because those are key as a very strong enhancer element to confer very, very high expression levels in the red blood cell lineage of anything linked to them. So it was a challenge. So that's the final vector that we used. I'm not going to go through the details if we don't have time, but basically you recognize here the beta globin gene with the mutation that I mentioned, the LCR elements within a lentiviral structure. Now, we made a lot of progress in animal models, mouse models of thalassemia and sickle cell. We published, for instance, in 2001, a proof of principle of correction of sickle cell disease in mice. And it took us a few years to move forward to doing clinical trials, but actually it wasn't that long, because now in 2016, which is like 15 years later, we have already transplanted the first patient 8 years ago, and we have now transplanted close to 20 patients. So it took a while, but things are moving forward quite nicely. And as I just said, it took several steps from colleagues and ourselves to move the field forward. 2007 for the first beta thalassemia major patient transplanted, and in 2015 we have almost 20 patients treated for both thalassemia and sickle cell, both in France but also in the US in multi-center trials with patients coming from Thailand, Australia, the US, and Europe. Now, the overview of the clinical protocol. First, the key eligibility criteria are for beta thalassemia major to be severe with at least 100 mL of packed red blood cell transfused per year, or severe sickle cell disease based on key criteria. And of course, the patients cannot have an HLA matched sibling donor, otherwise they would be candidate for allogeneic stem cell transplantation. So we harvest CD34 plus cells, either from the peripheral blood or from the marrow. We purify them and we expose them to the vector that has been thawed after being cryopreserved and analyzed. Then the cells, after transduction, are themselves cryopreserved briefly to perform some key testing. And if everything is fine, the patient goes back to the hospital, receives only one conditioning agent, busulfan, which is an IV form of busulfan, Busulfex in the US or Busilvex in Europe. And after a few days to wait for the washing period, the cells are infused intravenously and they home back spontaneously to the bone marrow of the patients. And the dose of cells we aim at is at least 4 million CD34 plus cells per kilogram. Now, in terms of conditioning regimen, we wondered whether we needed to use full myeloablation, even though we use only one agent, or partial myeloablation. Well, clearly, in terms of efficacy, there's no evidence in animal models that are convincing at the dose of cells we can inject back in the patient that it works at this stage. It may work one day, but so far it doesn't work. And in non-human primates, it's the same. Now, in terms of safety, it is not actually that clear that partial myeloablation with such an agent such as busulfan is safer than fuller myeloablation, because you may induce mutations in the genome of the stem cells, and since it's non-lethal, both cells do not die, and that could be a concern long term. Now, the first patient, as I explained, that we transplanted without injection of backup cells was in June 2007, and we published earlier result almost 3 years after in Nature, and we have followed the patient now for many years, over 8 years. This patient had a severe case of beta E, beta 0 thalassemia, so one allele was not making any protein, and the other one was making the so-called beta E variant. And because it was compounded, the patient was profoundly thalassemic with transfusion independence since early childhood. And we decided to focus first on beta E, beta 0 thalassemia patients because there are more and more of those patients through immigration from Southeast Asia in the US and in Europe. And in addition to that, only a few grams of hemoglobin in the blood are sufficient to go from not being transfusion dependent to being transfusion dependent. And therefore, we decided to see what would happen after gene therapy is performed on those patients. So, as I said, the first patient was 18 years old at the time. It has severe beta E, beta 0 thalassemia. He was transfusion dependent since age 3 with large volumes transfused. His spontaneous hemoglobin levels went as low as 4.5 grams per deciliter. He had major hepatosplenomegaly and was splenectomized at age 6. Hydroxyurea was not effective. The liver damage and other tissue damage coming from excess of iron through the transfusions was not high enough, rather was could warrant transplantation because it was not too high. He was there's only one child in the family, therefore there was no related geno-identical HLA matched donor, and he was a candidate for gene therapy. He was transplanted 8.5 years ago at age 19. Now, here I'll show you what we can measure in the blood. The amount of hemoglobin in the blood in gram per deciliter, and you see here the number of months after transplantation, and it keeps going until now, which is again 8.5 years post-transplantation. So you see here in different colors the different types of hemoglobin that you can measure. So at the top in light green, that's beta A coming only from transfusions. Then in red, it's beta 87 coming only from the vector. And you see here in blue and darker green, beta E, because the patient, again, is beta E, beta 0, and gamma, the endogenous gene that goes de-repressed. And as you see, you cannot detect after a while, which is about a year after transplantation in this patient. It was slow, now it's much faster, I will show you later. You no longer detect any trace of beta A because the patient is no longer transfused. However, you see the rise of beta 87, and it's stable with about 1/3 coming from each species, beta 87 from the vector, and beta E and gamma. And the patient is maintained at a very mild anemia of about 9 grams per deciliter in spite of regular phlebotomies to remove excess of iron, which the clinicians decided to do. So in spite of that, the patient is maintained. He received only 2 transfusions for comfort, but otherwise appears transfusion independent. Now, in terms of circulating erythroblast levels, they come down as expected. The red blood cell lifespan is also largely corrected. The internal content in hemoglobin is also now in the normal range instead of being way off in the beta E, beta 0 thal patient population. And we calculated that the gene expression output level was close to 100% in that patient. Now, as we published in Nature in 2010, we were a little bit concerned first because a larger proportion of cells than otherwise had an integrant in one locus called HMGA2. But nothing has happened since now almost 8.5 years. Actually, it is 8.5 years. And also, you can see here that the contribution of HMGA2 at the bottom has come down, and now it's no longer the most common site of integration. So that issue so far has flown away, and the patient has now polyclonal reconstitution with only a minor component coming from HMGA2. Now, we went back to the drawing board to improve upon this. And what we did first was to remove the insulator because the community felt that it was not warranted. There was no strong proof of concept that insulators were actually effective in this context. And we also purified by chromatography the vector much better. And we kept the beta 87 mutation, of course, for the reasons I explained earlier. So now, what is the status of the patients we treated? That's the status in December 2015. We have done more patients now, but they have not been disclosed to the public. As you know, the sponsor, Bluebird Bio, is a publicly traded company. Therefore, there are very strict rules as to what can be disclosed and when. So this is the status as the last December 2015. So in France, there's one trial in one center for beta thalassemia major and severe sickle cell disease. 7 subjects are planned. 5 have been already transplanted with a follow-up of at least 21 months, actually for the longest. We have 3 beta 0 patients, all beta major transfusion dependent, 1 beta 0 equivalent, which is the splicing mutation that is the most common, and 1 sickle cell disease patient. For the US, we have now the HGB204 called North Star study with 4 centers in the US, 1 in Australia, 1 in Thailand, for beta thalassemia major of all genotypes. 15 patients are planned, and we've already transplanted 11 subjects, 7 with beta 0 and 4 with Cooley's anemia beta 0. The longest follow-up is about 18 months. And that's, again, as of December 2015. And for the last trial, HGB206 in the US, also multi-center with 3 to 6 planned centers, all in the US. It's only for severe sickle cell disease, with now 20 patients planned after an amendment, starting with 8, now it's 20, and 3 subjects have been treated. The longest follow-up is 5 months. So what is the summary of the clinical outcome? So far, all the beta 0 thalassemia subjects, whether in France or in the US, have rapidly become transfusion independent. With regard to beta 0, the few beta 0 thalassemia patients, and also it's quite early, they have either become transfusion independent or shown a major decrease in transfusion requirements, and we hope that those will also become transfusion independent, but this is still pending. And updates were provided at ASH in December, and I will just show you here a few slides on the French trial, which is with the longest outcome and observation period for the patients. So now for thalassemia patients, for this new trial in France, you see here the proportion of total hemoglobin compared to, due to beta-87 compared to total hemoglobin, and you see that all the patients, the 4 subjects, are now in the 70+ range. So it's very high, okay? And that's months post-infusion. In terms of the breakdown as histograms of the various species of beta globin that you can, I think this is not working. Yeah. Yes, it is, well, it's somewhat working. That you can, okay, abandon this. So you see here histograms in dark blue, that's HbA coming only from transfusions. You have in green HbE for patients who are, beta E, beta 0 patients. In yellow is hemoglobin F, meaning gamma globin, endogenous. A2 is delta globin, endogenous, with only a trickle. And 87 in red, that comes only from the vector. So the important ones are A and E, so the blue and the red. So you see here the 4 patients observed. You see at the bottom and on top the 3 beta E, beta 0 patient, and the second one from the top is the beta 0 equivalent patient. So you see here that with time, the patients are no longer transfusion dependent for those that have been followed for the longest, and you see a total hemoglobin level for the female, which is the third from the top, at 11 grams per deciliter, which is within the normal range, or most of the normal range, and almost 8 grams of this comes from the vector. And for the one at the bottom, that's a male, he has over 13 grams per deciliter total hemoglobin, with about 10 grams coming from the vector. So these patients are doing well, and that happened very rapidly. They left the intensive care already transfusion independent. They never received a transfusion since then. And then the one at the top is earlier. It's 8.6 grams with about 4 grams coming from the vector, and we are waiting to see. For the beta 0 equivalent, it's 9.6 grams per deciliter with about 7 grams coming from the vector, and we are also waiting. So the results are quite positive. Now, if you look here at other parameters we could measure, parameters of markers of disc erythropoiesis, LDH, reticulocyte counts as witnessing hemolysis, for instance, those have been largely corrected. And in one patient for whom we were able to do a liver MRI, there's also an improvement in liver iron content. And the bone marrow smear looks normal. Now, in terms of sites of integration, we were pleased that it's always, so far, all the patients tested polyclonal. We don't see any dominance at any site, and it's quite a lot of various sites of integration. And the maximum single contribution is less than 8% of total clonality. Now, in sickle cell, so we observed there's one patient we have followed in France, the only one we did so far, and there again, the results are quite interesting, and we're going to publish that soon. At the post-transplant 6 months visit, the patient has been weaned completely from transfusion. You have to know that this patient who was young, less than 15 years old, this patient was on a transfusion regimen because he had a mild stroke or risk thereof. And we were able to wean off transfusion. I will explain why, because the HbS level fell way below 30%. There's no hospitalization, no acute episode since. The reticulocyte count has come down, and LDH levels have also come down towards normal values. And if you look here, comparing the TAL patients, the 2 with the longest follow-up, where you get about 70% presence of 87 in the blood of those patients, for sickle cell disease, it's rising, but we are already at 40% expression of the anti-sickling product, whereas we were aiming only at 10-20% if we were optimistic, because at that level, let's say 30 to be mild. So at 30% level, it's very hard to imagine that sickling would occur. Because again, beta 87 is a very strong inhibitor of sickle cell formation and HBS polymerization. So we are now past that level. So this is very encouraging. And if you look here at the breakdown of, again, hemoglobin levels in different species, you see here at the last bars on the right-hand side that in red, we have 40% expression of beta 87 in the sickle cell patient who does not have any thalassemia. And you get 5% HbF, which is also anti-sickling, but endogenous, and only 47% of HbS. So we're very pleased with this result. So the conclusions and prospect. Well, the safety profile is consistent with autologous transplantation without gene therapy or related adverse events and with polyclonal reconstitution. There's a reliable and robust conversion to transfusion independence of all severe beta E, beta 0 thalassemia patients, up to 10 grams per deciliter for HbA87, making 13 grams per deciliter total hemoglobin. And then we focus again on HbE beta 0 because it's the main current category of thalassemia major patients in the US and in Europe with, if you look at Thailand, approximately 3,000 new births per year of that class of thalassemia. Now, there are promising data in the first subject with severe sickle cell disease treated with gene therapy, production of anti-sickling hemoglobin at 45% at 6 months, well above the threshold that I mentioned, improvements in markers of hemolysis, no hospitalization for sickle cell complications for transplant, despite weaning off transfusions. And now for beta 0 patients, we see a major decrease in transfusion requirements or conversion to transfusion independence, and we're waiting longer to observe the patients and doing more patients. And we are fine-tuning current protocol to increase transduction efficiency for those patients, and we actually are filing an amendment with a new compound that increases transduction efficiency. And we have also designed, in collaboration with Jim Badolas in Australia, we have designed a variant of the vector that contains a microRNA within the second intron that triggers a decrease in the level of alpha globin, and only one of the 2 alpha mRNAs, so that the excess of free alpha globin is no longer present. We are optimistic to bring this to the clinic in trials very soon. And now let me just say a word about the cost of gene therapy, because one hears often that gene therapy is a medicine for rich countries and it may not be applicable where a lot of the patients actually are. Well, it may not be the case, because it is clear that during development and shortly thereafter, and we are at that phase, we need to recuperate the cost of development, and the cost is high. But it should come down, if proven effective, quite nicely and rapidly. And if you look at small molecules, the costs add up over the lifespan of an individual, whereas gene therapy, if it works, is a one-time treatment and that's it. And this is actually to compare the cost, why Thailand, which is a country of mid-range wealth, Thailand has decided years ago to reimburse the cost of allogeneic bone marrow transplantation for every child with beta 0 thalassemia that was transfusion dependent if they had an HLA matched sibling donor. And that's because the cost is so much cheaper than actually compounding the cost of transfusion and iron chelation over the lifespan of those people. And if you look at protein therapy, which also have to be delivered continuously for the lifespan of the individual, it's much more expensive. If you look at Gaucher disease, Fabry disease, and the variant thereof, the cost is almost at the level of $1 million per year per patient. And this is accepted and reimbursed in a lot of countries because you cannot let people not be treated if a treatment is available. So gene therapy, I predict, is going to be a rather cheap treatment of the long run, and there are ways also to help with reimbursement and coverage of those patients. So let's not go too fast. We are still at the phase of, proving concepts, but things, go nicely at this stage with more and more and more patients worldwide, and we hope that it will become a treatment modality in the near future. Thank you very much. Thanks all my colleagues in Paris, but also at Harvard Medical School in my laboratory there in Boston. And I'm also a visiting professor this year at the Mahidol University in Bangkok. So there are colleagues from there as well. And you have the clinicians and their colleagues, scientists in Paris at the Necker Hospital with Marina Cavazzana as principal investigator on the trial, Olivier Hermine, who performed the transplantation, Marianne de Montalembert, who is a great expert in sickle cell disease and thalassemia, who helped us a lot to provide patients, and our colleagues also in Germany and in Australia. And I should not forget Bluebird Bio, the sponsor, the company that actually produces the vector large scale and is the sponsor. Thank you.
Thank you very much, Dr. Philippe Lebouche, for your very illustrative talk, and now it's open for discussion.
On choosing the right vector, like the lentivector, to be introduced in the nucleus of the stem cell, even while in the quiescent state, the G node. Did you study the effect on the other lineages, the other progenitors, not just the erythroid? Are they being affected?
Okay, it's very hard to hear with the echo. The question relates to vectors.
Yes.
You're asking if there are other types of vectors, or if
Yes.
this type of vector can use...
Yes. You said that you've introduced this lentivector in the nucleus of the stem cell even while it is in the quiescent
Right.
state, in the G node. Yes, to import the gene. Did you study the adverse events on the other lineages apart from the erythroid? Were there any adverse events on the other lineages, like the myeloid, the monocytic, the megakaryocytic?
Well,
were they?
Right, okay,
Yes.
now I heard the question.
Yes, please.
Well, no, there are no adverse events in the sense that the patients don't have any side effects that can be reported, Again, we don't introduce the vector with our little hands into the chromosome of those cells. We just expose stem cell population to the vector.
Yes.
But what I said is that the vector has the capability of entering many types of cells, including the ones we want, which are the quiescent stem cells, And then after that, it stays there forever, it doesn't move, but it expresses, it transcribes into RNA, and you want transcription of the vector to occur only in the red blood cell lineage, and that's what it does. We cannot detect expression in other lineages. It's very, very strictly in the red blood cell lineage. I'm not saying that there could be some leak somewhere in some minor cell type, but it's so low that we can't detect that.
Okay. Second question, you said that the key eligibility were lack of donor, HLA matched donor, and just for beta thal major and severe sickle cell disease. I've noticed that you mentioned that there were no adverse events so far. Do you have any long-term studies or plan to study on other clinical types of thalassemias and diseases? As you said, now they are almost transfusion independent, no chelation. So why are you limiting the study on the severe cases only?
Well, we are not, because you want to do things progressively. Otherwise, you will be, or your sister, asking the same kind of question even more harshly, if we had not proceeded carefully, okay? So first you start with the cases where it's warranted, because the benefit-risk ratio appears to be favorable. That's what the agency wants you to do, whether the FDA or the NEA in Europe or local agencies. So then you go for patients who don't have any other alternative. because they are severe enough that they suffer. You see their life expectancy probably be shortened. They are in pain for sickle cell, or they have complications that are not nice. And at the same time, treatments such as hydroxyurea don't work for them. They are unfortunately in the subset of cases where there's no effect that is positive from that. And then they don't have an HLA-matched sibling donor, so you can't even transplant them with allogeneic stem cells. So you say, what do you do? So that's where you say, well, maybe it's warranted for them to try this, because we did, as you mentioned, long-term studies in many animals, many mice, a few monkeys, but many mice. And because of that, the agencies deemed it to be acceptable. And then you try first with a few patients. If it goes well, you try with many more patients. And then if one day, it works very well for those, you want to extend that to many more, and maybe even those who have an HLA-matched sibling donor, because the risk would be lower. You would not use, immunosuppression, et cetera.
Thank you very much.
Thank you again, Dr. Philippe, for your very illustrative talk. We are short in time. Sorry. Thank you all, and we come to the end of our session. And it was an honor to share this session with Professor Dr. محمد قاري. وما تنسوش بيفكركم بالبان أرب ميتينج في -3